And
thank you very much. And we move to the second speaker of the day, Mario Armelao from SCIEX, where he's a high resolution mass spec specialist. We got to know each other, I think, early this year when SCIEX was about to launch the new ZenoTOF platform. And I think that Mario will speak about this new instrument today. And I guess that there are a number of people in the audience who are pretty curious about this new instrument and what can be done in the field of lipid analysis. So I leave the stage to Mario. Thank you very much. And thank you to SCIEX for the support.
hello, can you hear me all right?
Sure.
And can you see my presentation already?
Yes.
I'll try to place this in presentation mode, so fantastic. So if you do confirm this looks all right, I will start by thanking you, of course, on behalf of SCIEX for the opportunity to speak at the event. And I would also like to acknowledge that the work that I'm presenting here was generated by my SCIEX colleagues at the R&D facility in Toronto, Mackenzie Pearson, Christy Hunter, and of course, the inventor of the new EED cell, Takashi Baba. And to kick off the presentation, following on Michael's topic, is really to acknowledge that analyzing lipids faces all the structural diversity that comes along with it. And just taking, for example, glycerophospholipids, we could see that the potential number of unique glycerophospholipids is really, really large. And this can even be further complicated by adding modifications. So if we have a look down at the sum composition level, so typically, and you can see by Michael's presentation at this level of identification, if we want to know the fatty acid composition, so this single P36-1 would turn out to have 5 possible conformations. And it's important to state that the notation at this stage will indicate that we do not know the positional level of each fatty acid. So if we want to know that, we need more complex technology, and that would further increase the number of potential lipid species. And in this case, we just considered one double bond. And for that double bond, depending on where it sits, that number further increases. And for each one of these double bond, it may depend if it is a cis or trans conformation, and this could have a biological impact. So from that P36-1, in the end, we have a whole bunch of potential individual lipid species. So in order to reach these level of identification, several techniques are really necessary. At the first stage, it's rather simple to go down to the composition level and even to the fatty acid identification. Just recently, I was reading a paper by someone in Stanford University with a 5500 Q-trap, analyzing triglycerides and doing the identification of each fatty acid chain. But in order to determine the fatty acid position, that is a bit more complicated. So typically we would stay over here, and in order to go down to the position, either some enzymatic type approach would be required, which ends up really downplaying the high throughput approach, or ion mobility, such as, for example, SCIEX's select ion differential mobility spectrometry could be applied. And down to the double bond, much more complicated, and to the stereochemistry, even more so. So the point is that currently there's no single commercial technology that can fully characterize a lipid, at the very least not in a high throughput fashion. And the real culprit of this is collision-induced dissociation. And okay, so in many areas, everyone can complain about collision-induced dissociation. It is the ubiquitous dissociation mode in mass spectrometry. But from proteomics to small molecule to lipidomics, everyone has some complaint about a given molecule that either does not fragment or does not provide sufficient diagnostic fragments. It's very fast, but it really lets us down often. And because of this, if we consider that the complexity of lipidomics arises from the fact that many lipids share the same precursor mass, many of those share the same fragments. So we see that even the MS evidence that we're collecting at a given stage would support a quantitative identification of multiple lipid species. So at this point, we acknowledge that, okay, collision-induced dissociation technology is quite limited. What can we do more? And this is where we introduce the way that SCIEX is now approaching characterization of lipid species, and that's with the new technology that we just launched this summer, which is the ZenoTOF 7600 system, which you can see here on the left. You can acknowledge this is a very compact benchtop system. It's QTOF based, as you can see the TOF, so it's rather small over here, and it's small because the flight path is doubly folded in an N-shaped design. One of the things I like to highlight because of TOF technology is that you could tend to suffer some mass stability issues if the lab temperature is not very stable. And the way SCIEX found out that this could be easily resolved was to, rather than try and keep the temperature from changing along the TOF path, we just decided to heat it up. So if we elevate the temperature above the room temperature, this is quite an easy, effective way to keep the TOF very stable with maintaining mass resolution and accuracy. And of course, this means we don't have to be injecting calibrant all the time. Just one comment, the ion source you see here is not the default ion source. This is the OptiFlow for microflow and nano configuration. So typically for lipidomics, most likely you would use the default Turbo V configuration. In this new system, you can acknowledge the QTOF design. I would say this is pretty much at the level of a 5500 triple quadrupole, with one exception, is that we have a new Q0 design, and this boosts sensitivity already. And as well that, of course, in this case, we use a hybrid ADC-TDC detector, which gives us immediately a higher linear dynamic range. But most importantly, we see the really fast acquisition rate. So this refers to a MS acquisition rate, and that's sampling rate. So that's counting all the times, which means that to achieve 130 heads, we're accumulating at 5 milliseconds at MS level. And you may think, okay, so 5 milliseconds is exactly the same accumulation time that is common to find in MRMs in triple quadrupoles. How can you do that at the MS level? And this is explained by the novelty part of the system, which I will highlight right now. So here you can see that in the section where you'd have a typical collision cell, in this case, you still have the collision cell here, as you can see. This is Q2. You can still do collision-induced dissociation here, but before Q2, you have a novel design. So this is a special ion trap, which is called a chimera ion trap, because it is a mixture of two designs. So in one way, it focuses and transmits the ions, and in the other orthogonal axis, it transmits electrons. So, and already you may think, okay, so free electrons, we have an emission source on each side, and if we're getting free electrons, the only way to maintain them trapped is to have a permanent magnetic field. And that's exactly what we have here on each side. We have a 1 Tesla permanent magnet. The other thing you may think is, well, it kind of sounds like ECD. How can I use this for lipids? So just hold that thought for later on. I would just like to say that because of the chimera design, it doesn't work necessarily as a classical ion trap nor a transmission quad. So it's actually a semi flow-through design. because the irradiation times are very short. So typically 10 to 30 milliseconds, which means that we can definitely use this type of fragmentation on an LC timescale using data-dependent acquisition. Being a QTOF-based system and SCIEX technology, we have no impact in terms of resolution and mass accuracy, just going at speed. Because the activation here is self-sufficient, we don't really need to continue to activate with the collision cell. So if we're using EAD, typically we're not using SID. And in this case, the ions would be transmitted to the last section, which is another novel ion trap design. So somewhat similar to a Q trap, this is a linear ion trap at the end section of the collision cell, but it's very special in the sense that it operates at the sampling rate of the TOF, and it does not scan. So it's a very high capacity trap, so much more capacity than the detector, and it does not scan. It merely lets the ions exit the collision cell in a way that they reach the pusher and the TOF perfectly aligned, increasing duty cycle in a QTOF-based system to almost 100%, so over 90%. In practice, that implies an increased sensitivity gain for example, the mass range of interest for lipidomics between 5 and 10 times. So if you can think you have this gain in sensitivity, you have this gain in specificity, so this really can compete with a triple quadrupole in terms of sensitivity for quantitation and speed as well. So for those that were thinking ECD, yep, exactly. EAD stands for electron activated dissociation, and you can operate at an energy regime that would allow you to do this type of fragmentation. So typically used for top-down approach, for example, or a little more energetic electrons would allow clear characterization of post-translation modifications. But for today's approach, much like the same for lipidomics, we would favor a much more energetic regime, so around 10 electron volts. In this case, the free electrons are captured by the ions, which forms an intermediate radical state, which is unstable and therefore fragments, producing charge fragment that we can analyze. So in order to achieve this high energy electrons, the reason why we can do this is really about the design of the ED cell and the capacity of accumulating a very high charge with it. And once we have this fragmentation, what would we want to do with it? Of course, we would want this fragmentation to allow us to achieve the head group fragment. We would like to fragment at the backbone so we can easily obtain evidence whether the backbone is a glycerol or a sphingboid backbone, and would like also to be able to determine the regioisomerism of the fatty acid chains. Ideally, at the end, we would like to be able to fragment the fatty acid in order to determine the double bond positions and ultimately determine the stereochemistry of that double bond, whether it's cis or trans. So starting from the first example, you can see here for the example of differentiating a sphingomyelin backbone from a glycerol backbone, you can see here we have the head group. And of course, because EAD can fragment differentially the two backbones, in the first case, so for a sphingomyelin backbone, we would have a single peak containing a nitrogen, and therefore it is an odd number single peak. However, if it is a glycerol backbone, we would have two fragmentation possible, so one retaining the carbon and the other the oxygen, and this doublet will signal unequivocally a glycerol backbone. Going further and complicating things, so looking at triglycerides, this would be the most extreme example of differentiating regioisomers. And for this example, I would refer to Takashi's paper that is published, and you can see the reference here, where they analyze this specific lipid. So it's the other way around here. So they know the lipid, and they're now going for the evidence. And the first thing you can see is that the SN2 position, it cleaves in two equivalent ways. So we have a single peak at the SN2 position, and we can determine the fatty acid chain attached at the SN2. However, for the S1, SN1, and SN3, although this seems dissimilar, it's actually the same, and you cannot differentiate the two peaks with some, and you have SN2 and SN3, but you can't really tell which fatty acid chain, in this case, is attached to the SN1 and SN3 for triglycerides, of course. Looking at the fatty acid position, one of the things that we can see, and I point your attention to the intensity right here. So the data that Takashi acquired was mainly in infusion-based mode because it didn't have Zeno. But now we have Zeno, so we can do this in an LC timescale. And here's an example how we can keep fragmenting the fatty acid backbone until we hit the double bond. And because of that, the loops of 14 Daltons become 12 Daltons, and we can assign directly where our double bond position is. So I know here is in the 4th position, which is uncommon, but that's exactly where Mackenzie found it. I really can't explain why that is. So far I already showed you the technology. I showed you that we can easily go to the lipid class, fatty acid identification. We can go down to the positions and identify the double bonds. But what about stereochemistry? So in this example, I'll go for, I think it's APC. And here you can see that the entire fragmentation, which is already published, you can see we have in the same spectra, the Ed group. We know it's a glycerol backbone because we have the spectral evidence. And we know that in this case, which is the fatty acid position at each SN1 and SN2 positions. Also, because of the fragments we have at the regime, we can tell the double bond position. And I would add as well that we can tell whether or not this is a cis or trans conformation. And to explain this, to show you this, and this is related, of course. So this is one of the most difficult structural entities to elucidate, right? And what we can see here is using the peak ratios generated by the fragmentation of the carbon-carbon single bonds. So next to the double bond can be used to determine whether this is cis or trans configuration. So specifically the methyl terminal side of the carbon chain, the radical fragment, and the hydrogen loss non-radical fragment have different intensities between cis and trans conformation. So in this case, you could see, again, well published, Takashi used two different conformations to measure and prove that we could differentiate the two conformations, both cis and trans. And with this, you can see that EAD at the speed at which it can be acquired and when the sensitivity provided by Zeno allows a novel, very high efficiency, high throughput solution for lipidomics. And I thank you so much. A little bit delayed and open for questions. Thank you.
Thank you very much, Mario. Super, super interesting. And looks very any question from the audience? You can raise your hand and unmute yourself. Now, then I have a question for you, Mario.
Mm-hmm.
I mean, it looks like now you can put together a lot of information for each individual lipid that is eluting out of a column down to the cis-trans isomerism. I'm wondering how you plan or how you already integrate all this information in the identification power. How does your software work? Is it working with, I don't know, ion libraries and you've got all this information available there and you assign a match? How does it work? How do you actually use all the information that you can generate?
Yeah, that's a very common question because, of course, we can acknowledge that the evidence is here. So we are acquiring, we have the spectral evidence, which is the starting point. And of course, being EAD, and let's say most of the systems are not were not prepared to process this type of fragmentation, that required a little bit of that adaptation. So we do have, when we are working both in-house and with third parties for releasing softwares for processing this data. However, I am compelled to wait for the ASMS meeting, which is right around the corner, for disclosing that information.
Okay, okay. Sure, no problem.
But it's of course the evident question, right?
Okay, okay. Any other question for Mario? No, then thank you.